Hi everyone, we're going to be talking about our follow-up experiment on engineering chlorsulfuron-resistant crops. So a quick overview of the topics covered in our presentation. We'll start with observations from previous studies and then move on to our research question. From there, we'll share our hypothesis, research methods, and a prediction based on those research methods. To wrap it up, we're going to be talking about the big picture and why our experiment is important.
So to start, these are some observations we have drawn from past research. The first observation is that in the enzyme assay of the mutant Arabidopsis plant, some enzyme in an extract interacts with the herbicide to prevent the inhibition of AHAS activity. However, there is little evidence of which enzyme is actually directly interacting with the herbicide. We have also determined exposure to chlorsulfuron at increasing concentrations decreases AHAS activity. We also know that we have an isolated DNA sequence that contains the ALS gene for the GH50 Arabidopsis.
So we observed that there is some enzyme in the GH50 Arabidopsis extract that interacted with the herbicide, but we don't know which enzyme specifically. Since we believe we have identified the DNA sequence containing the ALS gene from the chlorsulfuron-resistant mutant GH50, we plan on using this gene to test if the mutant AHAS enzyme is the enzyme that builds resistance to the chlorsulfuron. To examine this, we asked this question: how is the AHAS enzyme function impacted by the chlorsulfuron herbicide? And now our hypothesis for this research question. We propose that exposure of chlorsulfuron to the AHAS enzyme inhibits the activity of the enzyme because chlorsulfuron alters its structure so that it can no longer undergo a reaction with pyruvate.
So now I'm going to talk about the methodology we proposed to explore this hypothesis. So again, we saw in the Figure 3 experiment that in the enzyme assay of the mutant Arabidopsis plant, some enzyme in an extract interacts with the herbicide to prevent the inhibition of AHAS activity, but we are not quite so sure which one. So, our experiment plans on creating a sample using in vitro methods that only consist of the AHAS enzyme. This way, we can make a direct link between chlorsulfuron resistance and the AHAS enzyme. Our experiment is similar to Figure 3, but the key difference is that ours is narrowing down possible enzymes involved in this resistance, whereas Figure 3 used the entire protein extract from the plant. We would start by creating two AHAS enzyme samples, one made from the GH50 mutant gene from the Arabidopsis, and another sample made from the wild-type gene from the Arabidopsis. The experiment would consist of exposing both AHAS samples to chlorsulfuron at varying concentrations, starting with no exposure defined as a control, and then increasing concentration from there. We would then add pyruvate to each sample so that the enzyme can start its reaction. The specific activities of mutant AHAS and wild-type AHAS samples are then compared to determine if an appreciable trend exists.
So now we're going to touch on our prediction for the follow-up experiment. We predicted that as chlorsulfuron concentrations increase, AHAS samples will have higher specific activities when they're encoded by the GH50 mutant gene compared to those that are encoded by the wild-type gene. This means that we think that the AHAS enzyme itself is the enzyme directly involved with the resistance. If this prediction turns out to be true, our hypothesis will be supported. This trend is visualized by the graph, where the orange line represents the GH50 sample and the green line represents the wild-type sample.
So now I want to wrap up our presentation by talking about why our experiment is important. To answer the question of why this experiment, we have to look at our overall goal. This goal is to engineer resistance to the chlorsulfuron herbicide in multiple plants. To be able to do this, we have to first find a donor that exhibits this resistance, which we have done with the Arabidopsis. Next, we have to create a genetic construct of the gene that encodes for this resistance. This is the step we are currently on. If our prediction holds true, we will be much closer to identifying the exact gene that encodes for this resistance. Once we get this genetic construct, we can transfer this gene to a desired host crop, whatever that may be. If our prediction is false, then our hypothesis is refuted. meaning it is likely that another enzyme interacts with chlorsulfuron and not AHAS. This means that we would have to go back to the drawing board, which would involve finding evidence of other enzymes that could be involved in this process rather than AHAS. Thank you for listening to our presentation.